It's always fantastic to come and see you guys on a Saturday. I think it's brilliant that you've come here. I'm a scientist, and some of my science students have come today. Fantastic. I've already had a snoop at the posters. I've seen one on phage therapy and Pseudomonas, so if that's yours, I love it. I know there are some science students here from Sheffield as well, from Andrew Fenton's group. And I really think this is a valuable thing to do on a weekend because it brings us all together from our different disciplines. I am an antibiotic guardian, and I've signed up to try and come up with innovative problems to solve antimicrobial resistance. And I work with a lovely lab at University of Nottingham to do this. My colleagues are looking for natural alternatives to antibiotics, and we use a very particular method to do this. So we use bacteria to kill bacteria. And the bacteria that we use are friendly bacteria that are not damaging and not pathogenic to humans or animals. And they have been out there in the world since the world got started, killing other bacteria. And so what we are trying to do is add another line of defense that we could use as an alternative to antibiotics. Everything you've heard about stewardship is really, really important today. My job is to work in more the antimicrobials, but I want you to know I do stewardship. And I'm just going to jump off the stage for a moment and show you that I do stewardship. So this week, I had an eye infection. And my eye was pretty sore, actually, and I went for this week, right, from the pharmacy, which is not an antibiotic cream, okay? I went for a cream that was just going to palliate my symptoms and let me see if my infection would resolve, right? Okay, so I'm not just here telling you I'm doing it, all right? So I think it's really important we think in our everyday lives and our professional lives what we can do. So as a microbiologist, and I'm a scientist, that's why I give a big whoop for scientists, I really understand the health professions in animal, pharmaceutical, nursing, medicine. They're really, really important. But my contribution is to be a microbiologist. Now, I don't have any good ideas because I'm a human being, so I don't know how to kill pathogenic bacteria, but luckily, these bacteria do. And as you can see on this picture here, this is the Bdellovibrio bacterium that I work on, and it's attacking and invading into other bacteria, and it can kill them. And so it can do this, and it may become a new treatment for infections as antimicrobial resistance becomes more and more problematic, as you've just heard it already has. So one of the commonest problematic areas at the moment is in antibiotic resistance to bacteria, and there are a lot of infograms everywhere about how dangerous a problem this is. You've just heard from Diane, you've just heard from James. And what we are trying to do is bring something new, but it's a long road. So we work on killing gram-negative pathogens that actually attack crops, animals, and humans, and they include antimicrobial resistant strains. So we go after Klebsiellas, Serratias, Escherichias, Salmonellas, Pseudomonas, and they may have antimicrobial resistance genes of all these different kinds, giving them resistance to carbapenem antibiotics and beta-lactam antibiotics. Now, we get our antibiotic-resistant bacteria from QMC, which is our local NHS hospital, but we have studied these natural friendly bacteria that can kill a wide variety of these bacteria naturally. And what we're trying to do is take them out of nature and make them more useful. And we get a lot of support from funding agencies to try and do this, right? So the aim is to try and use friendly bacteria as a treatment for infections. Now, these bacteria are natural. They come out of soil and water. They've been there for millions of years, but we've never tried to use them as therapies. Bacteriophages, which you may know as viruses of bacteria, have been used as therapies, but we need as many different armaments as we've got against microbial infections so that we can rest some and use some. And the contribution of bacteria against bacteria is really powerful to see. So I'm going to show you two movies. The first one, top right, is E. coli being attacked by these bacteria in a liquid environment. Here they come. They attach to the E. coli when they find them, and they invade inside the E. coli naturally, and you see them do this in a moment, there they go. They round them up and they kill them. Now that's good, they can do that in liquids, they do that in ponds and rivers when you walk by, they're outside in Aston at the moment. They can also do this on a solid surface, and lower down you can see a movie by my colleague Carrie Lambert, which we published in 2011, which shows at the very bottom of the picture a Bdellovibrio arriving and attacking an E. coli, and the whole cycle of them killing off these bacteria. It invades, the E. coli rounds up, And the E. coli is now being eaten internally by these Bdellovibrio bacteria. It's dead. And those bacteria convert it into friendly bacteria. And out they pop in a moment. There they go, right? And they go off and they seek more bacteria and they carry out this process again. They attach to them, they invade them, and they kill them, right? Now humans, we don't know how to do that. It doesn't matter to them whether these bacteria are antibiotic resistant, they can do this. Now this is a really complicated thing because a living bacterium like Bdellovibrio bacteriovorus, the bacteria eater that we work on, has got 3,500 gene products, right? The pathogens they're killing have got 4,000 gene products. So there's an awful lot of biology going on there, and this is a complicated thing to use as a medicine. So we want to look at the efficacy of this, but we want to also use it in living bodies and not just see this as a curiosity in a test tube. And so that's a contribution the scientists in my lab are making to this. We have figured out with lots of other colleagues that there is a cycle of bacteria being invaded, eaten up, and converted into these friendly bacteria. And the question is, well, how can you use that in the body? So with lots of colleagues, we've looked at how these bacteria invade, and a really cool thing is that they make a molecular porthole, right? Now, nobody knows about molecular portholes in the outsides of bacteria, but here, colored in red, are the small Bdellovibrio bacteria. Here's an E. coli, and here is a blue porthole that this Bdellovibrio bacterium is making in the side of the E. coli. And the Wellcome Trust, to a big scientific funding agency, ooh, that was scary, have given us some funding to work with Andy Lovering, who works at Birmingham University, on these bacteria and how they make the porthole. And one of the things we're trying to do with Andy is to actually build portholes in bacteria without necessarily needing to use all of the live Bdellovibrios. Because if we can just puncture these bacteria with portholes, that may be a way of killing them. But also, it's amazing to see these Bdellovibrios invading inside the bacteria because they also eat them up. So here's a green labeled pathogen being consumed by dark Bdellovibrio, and it's actually eating up all of the nasty components of that pathogen and converting it to friendly Bdellovibrio. And that could be a good thing in a human infection. So the challenge for us is to do this for real AMR pathogens that make people sick. And so my colleague Jess, who's in the front of this photo, works with our NHS colleagues. You can see Matt and Gemma here, and they give Jess some of their worst infections that are causing problems in the hospital. Now, we haven't started with human patients, and we can't go straight into humans. So what we did was we collaborated with two fantastic scientists in London, Serge Mostovi and Alex Willis. And actually, Alex used to be one of my undergrads that I taught at Nottingham before she went down there. And we used a mock patient. Now, our patient is on the right. Our patient may not look like a patient, but it actually is a super patient to study. So our patient is a zebrafish larva. Now, zebrafish, as you may know, if you have any in tanks, lay eggs, and they develop when fertilized into larvae, and they make small little creatures. Now, it doesn't look much like a human, but it's got white blood cells, it's got a bloodstream, it's got most of the bodily compartments that a human has, and most of its genes have a homologue in humans, which is very, very interesting. So we have been testing our new treatments in these little zebrafish larvae because we can anesthetize them, we can test them with an infection, and then we can see what we can do as a cure. And this has been a fabulous way to look at if we put a pathogen, and the first one we used was Shigella flexneri, into the zebrafish and cause it an illness, can we then try and treat it with these living bacterial treatments? And we're able to count how many bacteria are in humanely sacrificed larvae. We're able to set up loads of these infections and look at how many larvae can survive the infection with the treatment. We can also make the infection fluorescently tagged, and we can see the infection develop inside the larvae, and we can see our treatment, which we can also fluorescently tag in a different color, develop inside the larvae and try and treat them. What we can also do in these larvae, which you can't do in people, is we can see the innate immune cells because you can label the white blood cells of the larvae in different ways and watch them. We can also switch off the production of innate immune cells to mimic a patient who might have a neutropenia. So we can make a larva that doesn't have any macrophages, for example, and see how the host immune system interacts with our treatment. Now, one of the things that I think we have learned through all the years in research is that there is a host component to recovery. And that is true in antibiotic treatments as well. We take antibiotics, we think the antibiotics are killing all the bacteria that are causing our infection, and we may get better if they're not resistant. Now, actually, what we don't know is that some of those bacteria may be actually being killed by the host immune system, not directly by the antibiotic. And because it all worked, we never actually worried about that too much. And so something that I really admire in my scientific and medical colleagues at the moment is people studying what's called resolution, right? So you start to treat an infection, but how much of the host bodily system then contributes to actually clearing that infection? You get the beginning of the treatment with the drug that you may have, the antimicrobial, or in our case, the predatory bacteria, but also there's a component of the host as well. And having a healthy diet, keeping ourselves in a healthy state is all beneficial to that resolution part, which is the host part. So in these experiments, we're able to ask all of these different questions about infections. Now, first of all, we had to treat with just our friendly bacteria to see if they were really friendly. So we injected friendly bacteria alone into the hindbrain compartment of the fish. And we had chosen that as a place we were going to set up our infection later. We were able to see them going in because we colored them red fluorescently. We're able to set up lots of these infections to see if any of the fish got sick and died, but they didn't. 100% of the fish stayed well when we had put the Bdellovibrio into their hindbrain. That was surprising, because you don't think about injecting bacteria into the hindbrain of an organism, but we were very lucky and our experiments worked, and the friendly bacteria were friendly in the hindbrain. What we also were able to do is count how many of the friendly bacteria stayed in the hindbrain. And up at 48 hours, there was still some friendly bacteria alive in the hindbrain. They were surviving very well in the early hours that we injected them into the hindbrain. Lovely. That means that they're alive, and so they might be able to kill any pathogens that you have as an infection in that site. And we were using this hindbrain site as one place to do our tests. Now, Serge and Alex had already established that if, however, you used a pathogen in the hindbrain instead, then you got lethal infections with this number of bacteria, 2 × 10⁴, and that the fish would be predominantly dead at 72 hours. And what you could see happening is you could see if you used a green fluorescent pathogen that you would get more and more green fluorescent pathogen growing over the hours in the brain, and that would cause the death of these fish larvae. So the Bdellovibrio don't cause any death or illness alone, and we have an infection that is otherwise lethal. So we decided to set up some experiments and have a look at this. And we could take movies and watch infections, and we could add the treatment. So if we set up the pathogen infection alone with just a buffer, what we saw is as the anesthetized fish was lying under the microscope, we could actually see inside its hindbrain this green growth of the pathogen. And it grows up over a number of hours, and it leads to a lethal level of infection. And just after this 19 hours that I show on the movie, the fish will all start to die. Okay? If, however, we had the pathogen, but we had a predatory bacterial treatment, so we have green pathogen and we add some red predatory bacteria, something very different happens over that time course. And what happens is that the green pathogen is attacked by the red predatory bacteria in vivo. The red predatory bacteria eat the pathogen, and they invade it and kill it, like you saw in the movies. And we believe that there is also a contribution from the host in wiping out the last of the pathogen, which it was previously ignoring when the pathogen was alone in a treatment that did not involve the predatory bacteria. And so we published this in 2016, and we were able to show that you could cure an infection in a living creature, and you were getting a single dose here of these Bdellovibrio bacteria, and we like to say that they were acting a bit like a fire extinguisher dose, right? You pumped them in, and they would extinguish the infection. Now, what we were also able to do was count the pathogens and see that they dropped if we had the Bdellovibrio present, and if we just had the buffer, they rose. We were also able to look at the survival of lots and lots of different zebrafish, and we're able to see that if you had just a buffer treatment, only 27% of the fish were still alive at 72 hours, but if you had treated them with Bdellovibrio, 67% were surviving at 72 hours. So there's a big difference when we had the treatment. We could check by looking deep inside their bodies because they're transparent, and we could see the Bdellovibrio invading and rounding up the bacteria and killing them, just like we could see in the test tubes. So it is that predatory action, that killing of bacteria that's going on, but it works in the body. Now, we also manipulated the macrophages, and we thought, okay, what happens then if we just make fish that don't have any macrophages? Are the macrophages playing any part in contributing to this clearance of the pathogen? And we found that, very interestingly, both the friendly bacteria and the macrophages had a contribution. We treat the fish with something called a Pu1 morpholino to switch down the level of macrophages. And when we do that, we see different outcomes in the larvae. Larvae that have got no macrophages at all do very badly with an infection with the pathogen, and they're all dead at 72 hours. However, even if they have no macrophages and we add Bdellovibrio, 25% of them survive. If, however, they are like the wild-type bacteria and they have macrophages and Bdellovibrio, 67% of them survive. So this showed us that we have got a treatment here that is talking to the host immune system and is live killing bacteria inside the living body. And this is a great thing to move forward with. So our first paper showed that the immune cells are working with injected predators to control that infection, which was Shigella, in vivo. Since then, we've been able to see that these infections that we can treat include Klebsiella pneumoniae, for example. Top row here, we've got red Klebsiella pneumoniae growing and buffer. On the bottom, we've treated them with dark Bdellovibrio, and you can see that the red pathogen here is not growing because the dark Bdellovibrio is treating it. We've also been able to look into the fish, color in the macrophages and neutrophils of the fish now, and watch how they are coming to the site where we might inject a treatment. And we can observe the behavior of the macrophages and the neutrophils, and we can see whether they're coming to the site to join in the infection treatment. And that's really useful for us. You can't do this in people because we aren't transparent and we don't have readily colorable macrophages and neutrophils, but we're trying to learn about what really cures a living creature. So this is really helpful, and we can count the numbers of leukocytes, white blood cells, that migrate into our sites. We've also seen something very cool. The Bdellovibrio actually interact with the white blood cells in the fish's hindbrain. So we've gone into cultured human macrophages and looked at that more. Here's a cultured human macrophage. You can see this also below in 3D. And this is some experiments we did with Dorina Ranganathan and Jess at our university, Jess Tyson at our university. And we could actually see that these Bdellovibrio-friendly bacteria could actually sit inside the human macrophages. That's interesting, right? And they didn't die. This was interesting. They died slowly. And so these bacteria can actually get into white blood cells as well as be circulating in the tissues and the fluids of the body where we inject them. Now, they do die eventually. 48 hours, their numbers have dwindled, and the macrophage hasn't ignored them totally, but again, they have a period where they're alive and speaking to the macrophages. So who knew that friendly bacteria could actually have a conversation with host white blood cells? Now, that conversation does not do anything bad to those cultured macrophages. They survive really well. And the predatory bacteria, the friendly bacteria, does not affect the macrophage viability. So this is good, but they're clearly communicating with them. Now, we also have looked at inflammation, because we didn't want to add a treatment that was going to make everybody really sick. So we have compared lots of inflammatory markers that you often see as high in an infection. And we used 2 infections with our cultured macrophages to look at these inflammatory markers. One was with a salmonella, which is shown in blue, and one with a Klebsiella, which is shown in red. And we measured inflammatory markers like TNF-alpha and IL-1 beta. We also treated with Bdellovibrio, and we compared what response we were getting from our macrophages. What we see is that the friendly Bdellovibrio are friendly. There is not a high level of inflammatory cytokines produced when we put the Bdellovibrio bacteria in, but obviously for the pathogens we do get a high level. So this again is good and encouraging. It's saying that macrophages know that the Bdellovibrio are there. They're not killing them really quickly. They kill them after a delay. The Bdellovibrio are available to have this conversation with the white blood cells, and they're present in the body. So we are now working to go from zebrafish further into humans, but here is a summary of where we are. So predation plus host response kills pathogens in live zebrafish, right? Surprisingly, inflammatory responses to the injection are low, but they do stimulate the host to tackle a pathogen load it's not able to clear otherwise. Bdellovibrio can take up a temporary residence in cultured human macrophages, but they don't damage them. After 48 hours, the Bdellovibrio are killed, but there is a time window of opportunity to treat live infections in the body. We've done a test on healthy human donors. We got them to give serum, and we've tested the serum for antibodies to Bdellovibrio. And we find very, very few people that have high levels of antibodies to Bdellovibrio naturally. This gives us the idea that we can give a dose of Bdellovibrio as a treatment. Now, we will not be producing an antibiotic of the future that you take over a weekly basis. We may be able to get a dose that is injectable and used as a one-off treatment for a serious infection. And so we are working to add that and get towards a treatment in humans with that we could add to conventional antibiotics as treatment. Eventually, a living body will raise antibodies to something that you inject it as a dose, like a living friendly bacterium. But this work suggests that we may be able to apply a dose of predatory bacteria in future medicine safety trials. Work really continues on this because we're in a new area. We have to establish that this is safe. We've done trials in chickens where we've given drinks of Bdellovibrio to chickens and looked at their well-being and growth. And they have natural and normal levels of growth and behavior, so they're not affected by that dose. But there is a lot of work to do on safety before we can get into humans, but we're doing it. And I am aided by these wonderful people in doing this work. I'd like to thank Jess, particularly, who organizes this side of our work. The US government, who do amazing research that you sometimes think of in very different ways, have been very stimulatory in this work. And DARPA at the US government gave us lots of funding to actually do this work to establish whether you could use Bdellovibrio in a living body. They've been amazing. They actually funded the Apollo Saturn rocket initially and the miniaturization of mobile phone components so that mobile phones could actually exist. And they've put resource into worldwide research on Bdellovibrio to get this towards possible treatment. Serge and Alex have been fantastic, and you are a community of people out here. Alex was my undergrad tutee. She used to sit in my office, and then a few years later we were doing these experiments in zebrafish. Chris Moore, who worked on our zebrafish project, and Darini, who worked on immunology, have been absolutely brilliant. We've had funding from the UK government via BBSRC and the Wellcome Trust funders at the moment to work on how you bore those holes into bacteria. So I'd like to thank you. I'd like you to think about your contributions. We all make different contributions in this area, and I'm really delighted that we're all here today, that you get an opportunity to chat. I'm going to hang around at lunchtime, so please come up and have a chat with me. I'm going to come and have a look at your posters in the lunchtime break. I do have to be gone in the afternoon, but thank you very much.